Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are wrapping up Chapter 13, which is viruses. So we've learned a lot about viruses, what they are not, what they can do. The last thing I'm going to cover in this chapter is prions. Prions are not viruses. They're something new. Remember we had said at the beginning that we said viruses are the smallest infectious agent, but I said to star it because we're coming back to that. Prions are smaller than viruses. So let's look up prions and see what they are and what they do. Okay, so prions are infectious agents. They are proteins. They're proteins that can spread a disease. And this is new. This freaked people out because we've talked about everything with nucleic acid, right? Those are the instructions. That's what makes the proteins. So you need instructions for things. So even viruses have some sort of nucleic acid. Even if it's not DNA, it's RNA, it's scaled down, but it's nucleic acid. Prions do not have any nucleic acid at all. They are only proteins. They can be passed from one infected person to another, and they can lead to diseases. And the disease that they lead to are a disease in a category called encephalopathies, which is involved as a degenerative brain disease. And it basically turns your brain into a sponge. So spongiform encephalopathies. Signs and symptoms for this include mental things. It infects nerve cells, so there's mental derangement, loss of muscle control. These diseases are progressive, so they get worse and worse, and there's no cure for them. They are always fatal. So this is a pretty big deal. Diseases that are in this category include mad cow disease, Creutzfeldt-Jakob disease, Kuru, and a chronic wasting disease. So let's talk about a little bit of this and what's going on with this. The protein is misfolded. Let's go back to the power slide. I'm going to do the next one. There is a protein that we all have called PRP. And we have it in our cells. It's part of our cell membrane. We actually don't know exactly what it does, but we know that when we don't have it or it doesn't function right, we get these degenerative diseases where your brain turns to sponge. So it's got to be doing something. We just don't know what it does. So all animals have it, all mammals, I think all animals, but all mammals for sure have it, and we call it cellular PRP. So remember, this is a polypeptide chain with amino acids. If you remember back to protein structure, it makes these alpha helices. So if it bends in a certain way, you get these alpha helices involved. And so this is the structure of this cellular PRP. And we know that proteins are very dependent on their shape. What they do depends on their shape. Okay, all right, so this is our normal PRP protein. There is a type of PRP protein which has the exact same amino acid sequence, but for some reason it doesn't fold in these alpha helices and instead makes these beta pleated sheets. And you can see that messes up the shape of this protein. So if it misfolds, and again, same amino acids, but it changes its shape and it folds a little differently, then it becomes a prion PRP. So it's a shape flip. So when this protein is in this conformation, it doesn't do its job. Whatever that job is, it doesn't do it, and your brain turns to mush. All right, so how is it infectious? If there's no DNA, how does one protein infect another? What happens is a person consumes this protein. You eat the animal that contains this prion PRP, which is this misfolded protein, and you ingest it. And we know catabolism, right? You break things down, you build things up. That protein gets in your system, and then your body recycles it and uses it and puts it in your cell membranes and uses it for your proteins. But this prion PRP isn't folded right. And so it gets inserted into your cells, and you have other normal, typical folded PRP. And when that prion PRP that's folded incorrectly comes in contact with a correctly, that's my alpha helix, we're going to pretend. If it comes in contact with this correct one and it's nearby, it causes the normal one to change shape. And now it looks like the prion PRP. And then if it's got another normal one on this side, this one will cause this one to change shape. And so it moves throughout, and as it's contacting these PRPs, it induces this shape change. And now those PRPs aren't doing their job anymore. So it can progress through, and as these prions shape flip and they change shape, and they don't do their job, the disease gets worse and worse. The side effects, the signs and symptoms, not side effects, the signs and symptoms of the disease get worse and worse until you lose all muscle control, you can't breathe, and you die. So they're always fatal. And again, targets nerve cells. So the things you lose first are your neural faculties, which is very, and then your muscle cells as well. Okay, so no nucleic acids, which sent scientists just crazy because we don't know of anything that can cause this without an instruction manual. So this is how these prion diseases work. The deal is this shape change and this one shape flipping to another shape and then causing another one to flip is a very long process. So you can be exposed to a prion and it may have a long period before you exhibit signs and symptoms. And that period can last for as many as 60 years. So you come in contact as a young man or a young person, and then 60 years later, you're coming down with the symptoms of this disease. And it's progressive and fatal. All right, so what was the deal with this? Mad cow disease was an example of this. There's quite a few animal prion diseases. There's bovine spongiform encephalopathy, which causes mad cow disease. A cow gets in, causes those damages in the cow. The cow, dies, but before that, it infects another cow. Or there were practices where you would take the cows that weren't going to be used and you would kill them and grind up the meat and the bone and then feed it as feed to other cows. So it could be spread from one cow to another because one cow would consume the meats from the dead cows. So this was happening in Europe in the 1980s, and they had an outbreak of this bovine spongiform encephalopathy. And the cows were exhibiting this mad cow disease, and they were having to kill the cows. Now, one thing about this is the only way to get rid of the prion is incineration, burning it completely. So incineration completely. There was no chemicals or anything, especially at that time period, that would get rid of these prion diseases. So they found this mad cow disease in the herd, and that was dangerous. It was an animal disease, but they found it transmitted into humans. And we know what happens after dealing with this COVID-19. There are lots of zoonotic diseases. There are a lot of diseases in animals that we don't get. But when that virus changes, or in this case, it's a prion, when something changes and then that infectious agent is capable of moving into a new species and causing a disease, then that's an issue. And that's what's happened in Europe in the 1980s. This mad cow disease, people were eating the cows from that. They had some herds that were infected, and then people were showing symptoms. And it took about a couple of years before symptoms started appearing, within a year to 18 months before the first symptoms started appearing. And then they realized what was going on. And now we've got these people that are infectious and have this prion disease, and it's a fatal disease. So at that time period, they had to kill herds of cattle, any then that had been anywhere in that vicinity. They had to kill those. Meat was banned. You couldn't import meat from areas in the United Kingdom where this was prevalent. So it was a really big deal. People have been involved. It's found out that it can transmit now. It came from animals into humans, and now we found human-to-human transmission. They found some transmission due to organ transplants. Had a donor that was infected and they didn't know it, and then transplanted parts of their eyes and other tissues into a healthy donor, and that donor got that spongiform encephalopathy. So this became a really big deal. A prion disease. And in fact, even today in the United States, one of the things, if you've ever given blood, one of the questions they asked you was, were you living in that area of Europe during that time period? And if you were, that excludes you from being being a blood donor because of the latency factor, because it could be decades before you exhibit symptoms. So you are banned as a blood donor for life from that. So this is an example of a prion disease. It's a little scary. Derangement, we said, very long latency period. This one, chronic wasting disease, is found in deer, and it's been found in deer in Native America, in United States area. We have not seen a jump between this one and into people from the deer population like we did with the cow population, but they are monitoring it. And so I think the hunters will call on, we're in Texas, a lot of people hunt zombie deers where they're exhibiting these neurological defects. And people are monitoring wildlife agencies are monitoring that to make sure that our deer population isn't passing this along. Kuru is an interesting one. This involves European explorers way back when and cannibalism, and then they came down with this encephalopathies. All right, so we understand how it happens. No nucleic acids evolved. It's only a protein being near there. We do have some protection. Cellular PRP, just like proteins, can have variants. Some people are naturally immune to this. They don't have a certain amino acid in a certain place, and then their cellular PRP is not susceptible to that shape change. So that's good. So 60% of the people are immune, but 40% are susceptible. So 40% have the type of PRP that is susceptible to this shape change. Again, we can pass it from human to human by tissues, transplants, blood transfusions, mucous membranes to transmit the disease. How do we get rid of it? Remember when I said with the animals, it only had to do with, all you had to do was incineration. That's a problem. Normal cooking, so if you have meat, you just cook it from a deer, that won't get rid of it. And that's how it spread from these infected cows into the human population. Normal cooking didn't get rid of it. Boiling it doesn't get rid of it. Even autoclaving, which we'll talk about autoclaving in a future chapter, but in the next chapter, autoclaving is kind of how you sterilize instruments. That wouldn't get rid of it. So the only thing that gets rid of it is this incineration. Now, since that outbreak in the 80s, chemical companies went to work and they found, created an enzyme that will target prions. So now we do have another thing in our arsenal to defend ourselves. But before that, all you could do was just complete incineration to get rid of it. So we do have an enzyme, it's called Prionzyme, that we can use for like medical sterilization, that kind of thing. This was the first time we saw cross species. Does this scare you? Are you now freaked out? I don't know how many of you are getting more freaked out the more we go into this course. Does it scare you? It's rare. It is very, very rare. The outbreaks we've seen have been contained. Again, that big one in the 1980s, that was a big deal, but it has been contained. They're monitoring it. The chances of you come in contact with having it are very low. And again, you have to consume that protein or a transplant type situation, which is monitor those screening questions. Were you in this area at any period of time? The wildlife management, managing it in animal diseases and watching it. We're watching it. It's something to be aware of. It's not something to be afraid of at this moment. But as with COVID-19, you never know when an animal virus is going to jump out. Chapter 14 is going to be great because we're going to talk a lot about that. Okay, that's the last thing in this chapter. Let me know what your questions are, and I hope you've understood a little bit more about viruses as we've gone through. Thanks, guys.